Good afternoon, everyone. Welcome to today's webinar. I'm Dr. Fan Chia-Ching from BNE NCKU. I'm going to be the chairperson for this webinar. Today's webinar is smart healthcare science and technology. Today's topic is the microsystem technology in tissue and biomedical engineering. So it's a great pleasure for us to have three of the world's foremost professors and researchers at the forefront of the biomedical engineering field. So let me introduce our first speaker, Professor Chia-Ching, who is sitting there, who is a professor and the chairman of Department Cell Biology and Anatomy College of Medicine, National Cheng University. Professor Wu has published extensively in NCI on the topic of tissue regeneration, stem cell, and mechanical transduction by nano-optical biotechnology. In 2015, he was awarded Wu Dayou from most of Taiwan. The second speaker, Professor Chamras, who is the assistant professor and advisor to the Dean of Department of Biomedical Engineering, Mahidol University, Thailand. Professor Promptmas has published several articles on the subject of microfluidic instrument, biosensor, and the micro-total analysis system. The third speaker, Dr. Pornpat Athamanolap, who is the lecturer of biomedical engineering, Faculty of Engineering, Mahidol University, Thailand. Dr. Athamanolap has published several articles on the subject of microfluidic instruments, molecular diagnostics, and infectious diseases. One of her recent studies has been published in Nature Communication. So before we start diving into today's talk, let me remind you, if any question crosses your mind during the presentation, you can type your question into the chat box. So let's welcome Professor Wu. He's going to share about his latest research. His presentation is titled Cell-Based Microfluidic Chip for Tissue Engineering and Regenerative Medicine.
Hi everyone. It's a great honor and pleasure to share my research work in stem cell and tissue engineering. And as Professor Fan mentioned, so I'm actually a professor in cell biology and anatomy, as well as joint appointed in our BNE department. Actually, I was graduated from this department, so I'm very honored to come back. And for the recent year, my research topic is focused on the cell-based microfluidic chip and how to create a microenvironment to help the cell to do the to make them become noise. Yep, that's the first speaker, Pavan. So usually we will clear some issue regarding to the technical issue. So I will just use the mouse to continue. Okay. So we know the stem cell is a very powerful cell. And the cell is sitting in different microenvironment, including with growth factor, scaffold, or many different factors. And then if the cell is cultured and keep in good environment, they can do the recovery or repair for regeneration. And for my lab, we recently focused on three different tissues in neural system, vessel system, and skin. So those are our publications. And besides a good microenvironment, we are also interested in, because in our human body, when we have disease, that means the microenvironment come out some problem. So we want to know what's the environmental transition, and how can they become an abnormal environment, and then will result in the different diseases. And then in the neuron vessel and the orthopedic, we also identify those disease factors and how they affect the normal, the endogenous cell. And then we are trying to develop some method, try to rescue them, to make them become the normal cell, and then to provide the repair function. So the first story I would like to share is the peripheral nerve system. And we know that we have the brain that can control how we can reach a thing, grab a thing, write the papers. And then, but the brain actually is sending the motor, the potential through this peripheral nerve. And then if we look into the details, then we have there's electro jump propagations. And this kind of jump propagation actually is formed by the swan cell, and the swan cell will wrap around just like our electron wire. They will cause some insulating property, and then the electron cause the jumping property. And the structure of those insulating material is the myelin sheaths. So the myelin sheaths is very important for peripheral nerve. If we don't have those myelin sheaths, or if we have a problem in myelination, that will cause all different kinds of problems. For example, in brain, that will cause Parkinsonism. And in peripheral nerve, they will become abnormal because your nerve propagation cannot pass through from the brain to the distal. And so in our lab, we create two different models trying to study different disease for this kind of peripheral nerve. And one is conjugate, which is pretty common in car accident or the work accident, because if you have a transplant work injury, like you have an amputation or the cut off your thumb or leg, we need to repair the nerve, and however the nerve is the most difficult to repair. So we have this kind of nerve conduit model to study how to promote the regeneration. The second thing is the compression, which is common happen or even in our young students, because we play a lot of computer games, and then after overnight play computer game, you will feel your thumb is thumbness. And then so the reason is because in our hand we have a carpal tunnel here, and if we have a compression of the nerve for long term, it will cause muscle atrophy. Now we call it actually is a carpal tunnel syndrome. And so we try to study the nerve disease for these two clinical problems. And the method we use actually is stem cell, and particularly we are interested in adipose-derived stem cell. The reason we want to use this one is because we can easily get by liposuction. That will make you become more beauty, and then we can also get more stem cells. And actually it's autologous. And we know that 2 years ago, Taiwan passed a cell therapy law. So now we can do the autologous cell therapy. So, but not only in Taiwan, actually it's all over the world. Nowadays, we have a lot of adult stem cell bank trying to isolate your own stem cell and keep it in the freezer, and actually it's a liquid nitrogen. And so when you are old, if you need to use it, you can use your own cell. So this is a normal cultural morphology of the adipose stem cell, and we are interested in one kind of material we call it chitosan. Actually, it's a chitin material. When we put the stem cell on the chitosan coated surface, actually it is a narrow coating, so the cell cannot attach to the cultural surface, and then instead they will aggregate together, become a sphere. And when we look into their immunofluorescence staining, we can know that within the sphere, actually it's a neural stem cell or mature neuron marker, but on the peripheral surface of the sphere, actually it's a GFAP, which is a glial cell marker. So we call this kind of the mixture population cell is a neural lineage cell, NLC. So which means that they have a mixture of the neural properties. So we create the rate with a cytokine nerve transaction, and then we have the cytosine conduit together with the neural lineage cell, and try to see if we can improve the regeneration or not. And the result comes out, if we use a current clinical use conduit, the regeneration actually is failed. So you can see at the two ends, they have a black dark tissue, which means that they have a nerve necrosis. Means that those nerves are failed to regenerate, and then they will become dead. So if you look into the myelin sheaths staining, you see no those neuron wire, and then so there's no nerve fiber, and also there's no those insulating myelin sheaths. However, if we use the cytosine conduit with the derived neural lineage stem cell, we can see many small insulated nerve fiber, and then and this is the electron microscope to see we have a very good remyelination for those injured tissue. When we look into the motor function, we can see this is just like our clinical patient. If you fail of nerve regeneration, you can see the hand is just like a cramping. And in mice, in rat, actually it's also the same. So the mice will have a very cramped foot, and then they will have an abnormal gait. Their muscle will atrophy. However, if we have a good regeneration, you can see their foot can open and they can work very good. And this is actually collaborated with Professor Cheng. And then when we look into their muscle, they retain the muscle mass. So it indicates those stem cells do pretty good function for nerve regeneration. So the next thing we want to do is to look into the mechanism, how those kind of nerve progenitor or neural lineage cell, they can have the repair function. So we look at the different days of the culture when they culture on the cytosine surface, and then use the Western blood to see the marker. Indeed, they are gradually increased all those different neural lineage marker. And in addition, we're also looking into their surface markers, which is FGF receptor. So we have all 4 different types of FGF receptor, and then we see when they form the neural sphere, their profile of those FGF receptor actually change a lot. And then we have an increase of the FGF-R2 and R4 and decrease of the R3. The reason we want to see all different FGF receptor expression is because we know FGF actually is a very important signal for not only the neural differentiation, but also for hair, for those endocrine systems. So and particularly we are interested in FGF9. So what we did is that we additionally give the FGF9 to those sphere. And what we found is that if we increase the FGF9 concentration, this sphere become they will form instead of form a big sphere, they will become many different small sphere. And when we look into their sphere marker, nowadays we found that they can switch from the mixed population of the neural lineage cell become the swan cell. And the swan cell actually is very important because when we mentioned at the beginning, those are the key cells that can form the myelin sheaths. So we are very interested to see how this kind of strong cell can do for the regeneration. And again, we use immunofluorescence staining to double confirm the expression. And then we also use the siRNA to knock down different marker receptors. And then indeed what we found is the R2 and R4 indeed play a very important signal, because when we knock down the R2 and R4, those small superior will aggregate together, become a big superior, and they cannot differentiate into the neural lineage. And those signal also is relative to the AKT and ERK signal. So we can, by those experiments, we can dissect that nowadays the FGF9 actually is through the R2 signal and to promote the AKT phosphorylation, and then they will commit those cells into the strong cell phase. And so the next thing we want to see is that if the FGF9 do a very good job, can we directly apply the FGF9 into the nerve? And the result is no, because when we directly inject those FGF9 into the nerve conduit, the conduit become a fibrosis. They have a lot of the neural scar formation, and then the mice cannot work. So what we did instead is we pre-treat those stem cells, make them become committed to the strong cell, and then we inject those cells into the conduit, and this is the result. So after this kind of the strong cell commitment, the FGF9 treated cell, they can promote even much better than the neural matrix cell. So you can see a very thick regenerative nerve fiber with very thick muscle mass. And in addition, we also try to know the difference between neural lineage cell and the strong cell committed cell. And what we found is that those strong cells, they can really participate into the remyelination, because we label those cells into the red color. So if the red color becomes a circle like this, so that means the cell, they can participate into the remyelination part. And then if we inject the neural lineage cell, they will become the peripheral cell, and they will secrete the paracrine to the anti-inflammation effect. And confirm that, we also knock down the SI, using siRNA to knock down the FGFR2, and then we found the therapeutic effect will be disappear. So what we have in short summary regarding to how we use the sterile stem cell, we can culture them on the chylosine surface, and then make them become the neuron superior. They will become a mixture of the neural lineage cell. And we also know nowadays that they will increase the FGFR2 and R4 signal. And if we directly apply those cells into the nerve, they have the anti-inflammation and paracrine effect. They can still promote the nerve regeneration, but through the anti-inflammation. However, if we apply the FGF9, we can make the sphere become smaller, and then those small sphere will become strong cell. And those strong cells can directly participate to make those wires for the insulating of the fiber, so they will become a myelinating strong cell. And the next thing is I'd like to share is that because we have the a very good micro and nano center in NCKU. So most of my microchip actually is done in our nano center, and they can have a very good feature regarding to the nano scale. So we can use it to create all different this kind of geometry for cell to manipulate the cell. So what we mentioned previously, if we coating the chylosine on the surface, they will become sphere like this. However, there is a problem because you can see those spheres are very uneven in size. So which means that they have different properties in neural lineage. So one of my students, they create this kind of chip, So we call it Pachinko project. The reason we say it's Pachinko is because that we know there's the cell is just like a small stainless ball. So if we have a bigger hole, we can recruit more cell. They will become a bigger size of the sphere. So we have those manufactured, and then we test the outcome. And then indeed nowadays, if we use this kind of microchip, we can create all different sizes of the sphere. And this is important because a sphere actually later on I will say it can be used in many different applications. So that's the story for NERD. And then I would like to switch the gear to the skin. So in recent year, unfortunately, we have several major injury and accident. One in North is a water park accident, and of course, a lot of people have severe brain injury, like more than 200 people. And then about 5 years ago, we also have Thailand, the building collapse, which is more than 100 people dead. And then in these two accidents, our hospital played a very important role in rescuing patients. So those are the acute brain injury patients, and sorry, it's a little bit scary. And then this is a flash injury for those patients. And then in those accidents, we serve several patients, and then we also use a modern technology. In here, there's a negative pressure device, and then turn out we can have them to closure a very big traumatic wound, and with a very good outcome. However, even when we use those negative pressure, we still know that this negative pressure wound healing device still have some limitation. So after that, we propose a new system we call real-time feedback negative pressure monitor system. And actually this is founded by our South Science Park, and then we also collaborate with University of Malaya. So this is the new system now we have. So we can, this is working in pig, so we can get a very good outcome to promote the wound healing, even better than the current commercial device. And then we also try to first diagnosis what's happening in the wound regions. So my collaborator, Dr. Pan, he also developed this kind of paper-based diagnosis system, which is they can directly diagnose or measure the blister fluid by in clinic. So to know the information status for patient, and then quickly we can how to treat the patient with proper drug. But however, the necrosis is still a problem. So we collaborate with Professor Chia-Ching. He developed this kind of nanoparticle, cubic that can carry NO. And NO is a very important molecule that can promote the angiogenesis. And so this PBNO can trigger by the IR laser, so to release the NO in wound. So we have, and then for us, we have this kind of skin flap, the disease model that established also previously. So we put these two models together and try to see if we can prevent the skin necrosis. And turn out the result is quite good. Now, if we inject those nanocubic into the skin and then shine a laser on the those probably will necrotic the flap, then we can rescue. So you can see the dark, which means the death of the skin flap. So if we can inject the nanoparticle, no, there's no effect. However, if we shine a laser, so there's a dose effect. The shine one time, you can rescue some. If you shine twice or three times, you can rescue more. And then the result is also coming very good that we can use this kind of the NO cubic to trigger them and to promote the angiogenesis. And the reason I want to study skin is because the skin actually is a very simple but very complex structures. They have all many different organelle and different tiny structure here. So if we want to do the regenerative medicine, definitely currently we see we can have many different kind of the regenerative vector. However, it's just like a Lego. If you put the Lego in a box, they will become just a Lego in a box. They will not become a very beautiful city like this. So you need to do something for those cells. So what we like to do is previously we see the circulation, and nowadays we want to see the hair. How we can use a chip to promote the hair regenerations. So again, this is a chitosan. So we use chitosan surface, and then we harvest the vice versa, the 胡须, the hair cell from the rat. And then if we culture the hair cell, the vice versa hair follicle cells, if we culture them in culture dish, the hair marker will dramatically decrease after 10 passage. But however, if we culture them in sphere, like using the chitosan surface, you can maintain their hair sphere property. And if you do the staining, you can see a very good hair marker, and you can see the sphere on the morphology in the microscope. So previously we think that maybe if we inject those hair sphere into the skin, we can promote the hair regeneration and then cure the ball of those hair loss for many people. But unfortunately, this kind of the sphere, they are not working alone to promote the hair neogenesis. So so we're looking into the detailed structure of the hair, and then what we found is that actually every hair root actually is surrounded by adipose tissues. So we want to know what the adipose can contribute to the hair regenerations. So one of my students, they used the adipose stem cell, and then they had the culture of the mixture sphere. And the second thing is that they mimic this kind of sphere, and then the adipose stem cell or adipocyte actually surrounded outer the sphere. So they have different kind of cultural protocols. So you can see the adipose stem cell actually is labeled in red color, and the green color is the hair markers. So if we have this kind of cultural structures, and step by step by the culture on the dish, and with the microchip, and then form the sphere for the hair cell first, and then they form the second layer, outer layer of the adipose stem cell. Then when we inject this kind of culture structure into the new mice, which means that they don't have hair, and then we can grow a hair for them. So this is the scheme. And if we inject the DP sphere, there's nothing. So that's a very interesting work. So nowadays we can have the no hair mice to grow hair. So the final part I would like to share is actually another the chip we developed for the kidney disease. And this is also the same as the we use the adipose stem cell, and we try to culture and help the cell to interact with other cells. So what we have is that we have the central part of the those culture the microenvironment, and the peripheral part actually is the kidney epithelial cell. So if we have this kind of co-culture, we can promote this epithelial cell to grow the hair. This actually is a cilia formation, which is very important for kidney epithelial cell. And then the cell will also become a cubic-like structures. So which means that those epithelial cells become functionalized. And then so we are thinking maybe in the future we can have many this kind of small unit and probably combined with the hemodialysis machine because the functionalize the kidney epithelial cell, they can secrete the factors, for example, like renin, those kind of factors to supply and to overcome the shortage of the hemodialysis machine. So those are my talk. So so this is the end of the and a small summary of my lab. So we start from how to repair, and then how to prevention the disability. So we try to make the disability disappear and then become ability. So but to have the tissue engineering or the organogenesis, and then later on to have the functional recovery. So if you're interested, you can, I will have any question, you can email me. And then particularly for those works, there's also still continuous happen in NCKU. So you're all welcome to join us because nowadays in NCKU we have many online programs, international collaboration, and lots of the conference. So I'd like to thanks for my current and previous member, and thanks for your attention.
Okay, thank you for Professor Wu. Thanks for your informative sharing. So now we are going to have a Q&A section.
Okay, so the question is, what's the color of the hair grown on the mouse post sphere injection? And then what's the implication? So in the sphere, the green color is the hair marker. We usually use HAY1 or the vesicant. These two markers to label the hair, we call the hair follicle. And so if the hair follicle they are compact and become a sphere, that means their hair function are good. But however, even their hair sphere function are good, they are not able to regenerate the total hair shaft. So we need to wrapping the hair sphere with the stem cell, which is labeled in red color. So if we wrap it, the stem cell alter the sphere surface and then inject it together into the mice, then we will see those dark posts. So that's a hair shaft. So those hair shafts is a hair. We can see just like if you pull your hair, you can see the root. That is a black color root. So that is the hair root for and you can see by eye. Okay.
Okay, one question from mine. I want to know if we use the stem cell to treat disease, how long will it take to have therapeutic effects?
Okay, that's a very good question. So nowadays I also collaborate with many companies, the stem cell therapy company, and then but what I would like to address is that the stem cell is not 100% perfect. Sometimes after you inject, actually mostly that will happen is that they will disappear within 2 weeks. That's for mesenchymal stem cell. So they're just like they are the army that they can help to clean out or wrap out or inhibit the inflammation, but they will also die together with those battles. But however, if we are talking about not the mesenchymal stem cell, if we can find them or reprogram or guide them become the progenitor cell, different tissue progenitor cell, so they can have the and then injected it into the those injured tissue. They can have more function to repair and retain inside those injured tissue. So this is, but this part currently is not approved by our TFDA yet, because for our cell therapy, we can only use the minimized manipulation. But this is a future to precondition those stem cell or to use a stem cell to become a functional unit. That's the future goal. So later on, I think next year, our Ministry of Health, they will announce we call cell therapy 2.0 version. So that's that. In that case, we can have some manipulation for the stem cell. And also, the second issue is that you can use for example, like exosome from the stem cell to treat the patient. That is also can be allowed in the future.
Okay, we thank the Professor Wu again, and we are moving to the next speaker, the Professor Promptmas. Are you there?
Yes.
Okay, Professor Promptmas, his presentation is entitled Cell-Based Molecular Chips for Biomedical Diagnostic Application. Okay, please.
Thank you very much, Mr. Chairperson. It's a great honor for me to have the opportunity to present our work on cell-based microfluidic chips for biomedical diagnosis applications. Today, I'm going to present on the microfluidics platform for detection of specific antibody. In this case, we will focus on the detection of myelin, oligodendrocyte glycoprotein antibodies. This is one of the projects of the example of our collaborations between the university in Thailand, starting from the initiative ideas from the physicians from the medical school. We have engineering school and medical technology school from Mahidol universities. Collaborating with the Thai Microelectronics Center of National Science and Technologies Development Agency, Thai German Graduate School of Engineering of King Mongkut University Technology in North Bangkok, and also Nanotechnology College of King Mongkut University Technologies, Rajabang. So the initiative idea starting from the requirements of new diagnostic platform for neuromyelitis optical spectrum disorder, in short, is the NMOSD. It is rare condition of the autoimmune disorder affected to optical nerve and spinal cord. The critical features and later imaging characterization of the disease are similar to those of clinical features found in multiple sclerosis or MS. So NMOSD conditions need to be differentiated from MS because many common drugs used for MS medications can lead to the complication in NMOSD. So, in the case of NMOSD, we found the antibodies to the NMO antigen on the cells. So normally we can detect the antibody in the case of an NMOSD around the titer of antibody around 1 to 10. or higher titer. So cells-based indirect immunofluorescence assays has been accepted as a reliable platform for myelin oligodendrocyte glycoprotein antibody. In short, I also call mock antibodies. The major pain point of this traditional cell-based immunosensor consume the large volume of cell sample and reagent resources. Moreover, the technique needs to be performed freshly in the cell culture laboratories. So our solutions to improve the laboratory diagnosis service is using the microsystem that integrate the several parts of laboratory functions into a single chip of small size as a few square centimeter size. The objective of this work is to develop the chip platform for enable antibody detection using cell-based and membrane-based surface preparation in microfluidic platform. So the microfluidic device will be, our market is starting from the requirement of two hospitals. The first one is, the requirement is around 600 chips a year for the last hospital and 1,500 chips a year for neurological institute in Bangkok. So our work can be classified into three different work packages. The first one, we work on the design and development of serological dilution microfluidic chips. The second one is to design and development of the microfluidic chips for cell immunoassays. The last one is to develop the immunoassay signal detection for the microfluidic chips. Today, I'm going to focus on the first two work packages. In normal practice of immunoassay method, for antibody determination, the sample in this case is the patient's serum will be processed by dilution of the sample using fold dilution techniques. For example, two-fold dilution or 10-fold dilutions depend on the concentration of the antibody. In our case, we use two-fold dilutions. And normally, we work on the microtiter plate. Each dilute sample will be applied into the location of immunoassays. The amount of antibody in sample can be identified as the highest dilution ratio, which presents the positive signal of immunological reaction. In practice, the serum is diluted in microwell plate, typically 96 well plate at multiple concentrations. This conventional technique is time-consuming and requires a skilled operator and precise tool in order to obtain the accurate dilution sample when the serum is diluted at very low volume concentrations. So this is the chip that we aim to design, the chips that can deliver multiple serum dilution in parallel from 1 to 2 to 1 to 128, which are the volume concentration ratio between the patient serum and the diluent. In this case, we use the phosphate buffer saline solutions. As we know, at very low Reynolds number, the liquid flow behavior in dominant, while the viscous force, so the fluid flow in the micro channel sheet is entirely laminar. It results in the difficulty of mixing a fluid sample with the buffer in the microfluidic channel. In this case, the mixing can be created by two different methods: the active solution or active mixings of the solution and passive mixing of the solutions. The active one has external force, to involve the promoting of mixing. The passive one employs the geometry specific channel to induce the chaotic advections. So, in our case, we decide to use the passive micromixer. The fluid transport can be achieved by the two mechanisms. The first one is the advections mechanism, where the bounce movements of two liquid streams is driven by the external applied force, such as pressure-driven flow. And the second one is the diffusion mechanisms, which is driven by entropy. The study will focus on two different fluids. In this case, is serum and buffer solution, with different viscosity, densities, and concentrations. The curved channel can be used to introduce a pair of powder rotating secondary flow, which are known as these vortex. So our work will start with the simulation for designs of the microfluidic channel for diluting of the samples. The final volumes-based simulations, in our case, we use the computational fluid dynamic software. In our case, we use ANSYS fluids. And also the simplex algorithms is used for pressure-verse decoupling schemes. Here is the one of the 7 micromixers are designed. Use the shape of the serpentine microchannel. Is alternative convex semicircle. Curved channel consists of 26 quadrant and 56 semicircle. The prototypes of this design, is fabricated but using the polydimensional siloxanes. Here show the result of the designs of the microfluidic channel for diluting the serum sample. You can see it consists of so many sample and diluent solutions. Inlet port occupies more space on the chip surface. And this system also requires a large number of fluid pumps to flow the liquid in each channel of fluid. To create, this is will create some difficulty in their operations. To solve this limitation, we make a new design by using the CFD simulations and also the simplex algorithms. So this is the new design of our chips containing 7 serpentine microfluidic channels. There are 2 volume flow rate control. Okay, this one and another one. This volume control flow rate is for the serum sample. They have 1 inlet of serum sample and separate into 7 outlets to the microfluidics mixer. And another network of the fluid flow is the direct, in this case is phosphate buffer. Okay, 1 inlet phosphate buffer and separate into 14 outlets to the micromixer pan. After the design by simulations, we decide to fabricate by using the facility of Thai Microfluidics Center facility that can fabricate by using the production of the silicon mold and imprint the PDMS dilution chips as the simulation results. This one is the chip was verified by the performance of the chip in the accuracy of the dilution of the samples by using the UV visible absorptions. So the result is very good on this table. You see, this is the target ratio calculated from the dilution ratio here, and this is the experimental results come out. It's almost the same figure here. And the sample, this is the example of the serpentine micro channels. Okay, and this is the confocal laser scanning microscope to show the well mix of the solution, 2 solutions. Okay. And the signals, okay, that is the immunofluorescence signals corresponding to, is well corresponded to the amount of the antibodies, okay, in the systems. So the second one is, I'm going to focus on the development of cell-based immunomicrofluidic chips. This work aims to develop the microfluidic platform for NMOSD associated antibody, in this case is MOG antibody detections. The platform can significantly scale down sample and reagent volumes required in the system. The target protein of NMOSD antibody will be immobilized on the microfluidic substrate via the cell-based and membrane-based preparation in order to ensure that the binding function of the target protein in microfluidic platform. In this case, we use the GFP proteins MOG, mock transfect humans, embryonic kidney cells, egg cells. it shows the adhesions. This is the result of the immobilization of the hex cells on different surface modifications. The top one is the starting point that we inoculate the cells onto the surface of microfluidic chips. Left one is the chips surface modified by using this one is the unmodified surface, and the right one is the modified surface by polylysine modifications. You see the cells in the surface with polylysine modifications. The distribution of the cell is well distribution comparing to the non-modification of the surface. And the number of the cells after counted for 48 hours, the number of the cells is 5 times higher number than the surface without any modifications. And the fluorescent signals. So this is the result of GFP, MOX transfix hex cells. It shows the adhesion of the hex cell in the oxygen plasma activation microfluidic channel with and without microfluidic modifications. And the fluorescent signal here in the top one is the surface without modification, and the lower one is the modification. You see the clumping of the cell here, and this one is well distribution, and the signals of the fluorescent signal of the protein. This one is higher than this one. Okay, obviously higher than this. So the integration of cell-electron dilutions, microfluidic chip and cell-based. This is the diluting microfluidic chip and the cell-based microfluidics platform. The combination of these two platforms was used together to evaluate the serum sample from the patients. We used 20 serum sample from the NMOSD patient, and another 50 healthy blood sample as a negative control. We found that the platform offered the optimum cut-off level to distinguish the normal and abnormal level of NMOSD patients, that is 1 to 4 and 1 to 8 dilution spectrum. So in conclusions, I'd like to conclude that microfluidic chip between of the NMO antibody detections consists of the cell-electron dilution chip and also cell-based immunoassays microfluidic chips joined together. The dilution in parallel PDMS microfluidic chips with 7 serpentine micro-mixing channel can deliver 1 to 2 to 1 to 128 volume ratio with a high level of homogeneity. The cell-based microfluidic system has improved the immunoassay performance of MOG antibody detection, and the integration of the serological dilution chips and cells-based immunoassays microfluidic chips promote the improvement of the immunofluorescent assays and reduce the turnaround time for laboratory service. And the last one is the microfluidic for serological dilution is the universal platform that can be applied into other immunoassay system. So this work is the work from different organizations, different institutes. So I'd like to thank. First one is National Science and Technology Development Agency, and also the Faculty of Medicine, Silas Hospital, to provide us the research grant from this project. I'd like to thank the Royal Golden Jubilees program for providing the Ph.D. scholarships to work in our projects. I would like to thank all the people involved in the project from Thai Microelectronics Center, Thai German Graduate School of Engineering of KMU TNB, and also Nanotechnology College of Cheng KMU TLB. And lastly, I'd like to thank all of the audience in this seminar. Thank you very much.
Okay, thanks to the sharing of Professor Promptmas. So we have a Q&A section for the audience, please. Yeah, please.
Okay, thank you, Professor, for your presentation. I would like to ask, why did you choose serpentine microchannel as the shape of the microchannel in your research?
Yeah, first of all, we choose the method of mixing. The first one is the active mixing, and the second one is passive. We choose passive mixing. The active mixing is involved in a lot of hardware involved in the system. But for the passive, is even it look like a little bit complicated in design, but involved in a small part of the hardware involved in the flow system. And the serpentines have different designs of the geometric designs for introduce the turbulence of the fluid flows. Okay, so we tried several methods, even as the square, okay, the right angle turn, okay, and also using of the circular movements, okay, that is the spirals, okay. So we found the serpentine that is composed of semicircles connecting several semiconductors connecting can make the vortex flow better than the others by the simulation. That is our result from simulations.
Okay, thanks to the sharing of the Professor Promptmas. So let's move into our last speaker, Dr. Athamanolap. Her presentation is entitled Microfluid Device for Biomedical Application.
All right, can everyone hear me? Okay, good afternoon, everyone. I am Pornpat Athamanolap from the Department of Biomedical Engineering, Faculty of Engineering at Mahidol University. So first, I would like to thank this webinar series for inviting me for this event and for giving me the opportunity to share my research to all the audience. I'm very honored to be here today. And the work I am going to discuss in this talk is what I have done while I was doing my Ph.D. at the Johns Hopkins University. The topic is molecular-based microfluidic devices for biomedical applications. We have a digital microfluidic platform, and the two applications that I am going to discuss include bacterial identification with antibiotic susceptibility testing and evaluation of tumor heterogeneity from liquid biopsies. And the outline today will be basically the introduction with and then the design of our device and also about the cancer topic. All right, let's begin with the introduction of bacterial infection. It can happen in many parts of our body. It can range from even no symptoms, but you can infect other people or minor illnesses such as sore throat, ear infection, or the middle range like urinary tract infections, or the worst case of bacterial infections like sepsis, which is basically your immune system starting to attack your own body. That could lead to tissue damage, organ failure, and death. It is the third leading cause of death, account for 1 in every 2 to 3 deaths in hospitals. So it's pretty high. And when you have this kind of infections and you go to see the doctor, before the doctor can prescribe you any medicine, the information that doctors would like to know is, first is what type of bacteria that causes infection. And the second is what drugs that would work for each particular patient, as different patients can have resistant bacteria to different types of antibiotics. The gold standard of a current diagnostic method is still the conventional growth-based culture, which has been used for actually over 100 years. This conventional diagnostic method requires days to weeks to deliver the results. It consists of basically the gram staining, and then you grow it overnight, and then observe the result with the microbiologist. In the meantime that the doctors do not receive informative results, they are then forced to prescribe broad-spectrum antibiotics that may work against a wide range of bacteria. This leads to so many causes of antibiotics prescribed per year worldwide, not only in the US, right? And in the US, there are some estimates saying that nearly half of these prescribed antibiotics are unnecessary or inappropriate in selection, dosing, or duration. And to intensify this problem, there are also superbugs, which means is the bacteria that basically resistant to antibiotics. Antibiotics cannot kill them. And this is a global health issue because, for example, in the US, according to CDC, there are drug-resistant infections, about 2.8 million illnesses, and cost the U.S. about $25 billion in excess direct health expenditures. In Thailand, we spend about 10,000 million baht per year. There are 100,000 drug-resistant infections a year and resulting in almost 40,000 deaths. It is predicted that these resistant infections will kill 10 million people per year, which nearly 2 million people more than the cases would die from cancer. So if nothing is done, we are at risk falling back into a post-antimicrobial era, which means minor infections will become life-threatening. Are there any alternative diagnostic methods or platforms? Yes, there are. For example, Selfie Gene Expert, they have different panels for various types of infections, or bioMerieux Whitetech, where they have machines to detect over 300 pathogens, different phenotypic AST tests. There are plenty of platforms available, but none of these can replace the goal-based culture yet. So if we compare pros and cons of these diagnostic methods, basically the molecular diagnostic is fast. You can get the result within 1 hour or 3 to 6 hours. But for the identification part, the problem is they may not broad enough. For example, if you can detect these 5 things, but the patient infected with another type, then your 5 things will fail to detect the new one. And for AST, you definitely need to know the markers in advance. And there has to be relation between this gene and the phenotypic of resistance of the bacteria. And this provides limited information on the cellular response. And also this resistant mechanism keep improving. They are improving all the time. So it's kind of difficult to keep up to find the resistant gene. In the gold standard method, they are definitely good that it provides definitive results, ID and AST, direct measurement of cellular response. But the limitation is that it is time consuming. And why is it time consuming? If you're looking at how they do it, basically you would see that they have the plate, right? That starting from the left one, it is a blank plate, and then you go then for some amount of time then until you see the bacteria growing to this the middle one. The question is that how long does this process take for us to see this dot? And this one small dot, how many bacterial cells are they? Actually, this dot is not one cell. So it is we call them colony because they could contain about 1 million or billion cells of bacteria. Therefore, the bottleneck, how long does it take, could depend on the species of the bacteria. But the bottleneck of this method is definitely because our eyes are not sensitive enough to detect if they multiply from 1 cell to 2 cells. We need them to multiply from 1 cell to 1 billion or 1 million cells for us to see. Therefore, we want to improve or make this process faster. What we do is we propose a microfluidic device called nanoarray for bacterial ID AST. Basically, we will do broad-based identification and phenotypic AST. This figure shows like the schematic of this picture shows the schematic of the overall work, but I'm going into the detail of each step. Basically, what broad-based bacterial detection means, we utilize PCR by using universal primers. This means because bacteria have 16SR RNA gene, which contain both conserved sequence and variable sequence. The conserved sequence meaning all the bacteria have this part of the sequence, and variable meaning it is species specific. So to identify the species, we design the primers such that they are in the conserved region, plus the variable region that is species specific. So when we analyze the PCR product, we can identify the species. And the method that we choose to use to analyze the PCR amplicon or the PCR product is called high resolution MELT, or HRM. It is basically a pretty simple and fast method to investigate PCR product is in the closed tube system by monitoring a process of speed denaturing of double-stranded DNA in the presence of intercalating dyes. Different sequences will have different thermodynamic profiles that yield unique melt curves. And here we typically visualize the melt curve in negative derivative form, or we call them the melt peak. And what we could do is that we could couple this method with the broad-based PCR. So we could use this HRM for the melt curve-based classification, meaning if we know it's organism A, the melt curve will look like this. Organism B will melt like the orange curve. C will look like the purple one. It seems pretty straightforward. But in the case when we have multiple, we have big database. There's one example of 37 clinically relevant bacterial pathogens. So you could see that if you want to compare our curve to this database, it will become impractical because the curves are pretty close and it will be difficult to compare them. So this work we use broad-based PCR with HRM, and we have developed an automated identification algorithm using machine learning. This time we use the pork vector machine. And the flow is basically from the qPCR machine, we got the unknown melt curve. We input them into the computer that have access to our database. And the database has known melt curve of different bacteria. So and many more, like for example in this case E. coli is the orange one, Staphylococcus is the blue one. It will compute on what is this unknown melt curve are closest to and tell us, okay, this unknown one is highly likely to be E. coli. So this is the identification part. How about the antibiotic susceptibility testing? As some of you may know that after we do PCR, the bacteria already die, right? We need to analyze the DNA in the bacterial cell. So here what we propose is called phenomolecular AST, where there's, for example, we starting from the bacterial cell, we split them into two portions. One portion we add antibiotics. And with brief culture, if the bacteria is susceptible to the drug, the one without drug, then it will keep multiplying, it keep growing into your reaction. While the one with antibiotic on the top, the antibiotic suppress the growth. So different amount of cell can be observed after the culture, which reflect to different amount of DNA. So if we quantify this DNA, we can obtain the antibiotic susceptibility profile of the sample. Like for example, this case, it is the more the bottom one without antibiotics has more cells, then it's amplified earlier in the qPCR, while the one with antibiotics, then it will amplify later because it has less amount of DNA to start with. When we compare the CQ value of them, then we could say that this sample likely to be susceptible. And in this case, if it's resistant, then antibiotic cannot suppress the growth, then they should grow about equally. When you compare the CQ value, then they could be very close, and we can say that this is resistant strain. So in this work, with this workflow, we have shown that we can do the blind test where we start with a unknown sample, going to the phenotypic growth, broad-based PCR and SRM. We could correctly identify the species and also the antibiotic susceptibility profile of the bacteria. But then the challenge comes when if you have polymicrobial sample. Polymicrobial sample meaning you have more than one species in the sample, showing in different colors here. So what happens if we do the same method as just mentioned? The milk curve will show just the ensemble milk curve from all the species, which will be really challenging to resolve. What we propose is to do digital dilution, which means you basically dilute all these pieces of bacteria into large number of reactions so that each reaction contains 0 or 1 DNA molecules per reaction. And after PCR amplification and SRM, you will obtain individual milk curves, which can be, that means multiple species can be identified and also quantified. So this can be done using our nanoarray or the microfluidic device. This device can run about 15,000 reactions in parallel, and they are, the main advantages are the high throughput, the shortened assay time, less reagent consumption as each tiny small square reactions use about 1 nanoliter of reagent. It improves detection sensitivity, and importantly, it enables polymicrobial detection. So this is the structure of our device where there's a glass with PDMS. The main part is the outer thin PDMS, the channel layer, where it effectively prevents evaporation through the permeable material PDMS that we use here. And when we fabricate the chip will be lubricated to produce a negative pressure differential across the inlet and the outlet. So when punctured, the sample will automatically load into the chip, which then we can partition them with the oil after. At the end, you would see the green square, which means it's the reagents that are isolated into the small well. Then the workflow would become the same as previous, that we split the sample. Note that the initial sample you would see 2 different colors, the red and the blue, mimicking 2 different species of bacteria. And then when you load the DNA into our nanoarray to digitize them and perform digital PCR and SRM, we obtain the fluorescent images. With the ID part, then we extract the digital milk curve from all the fluorescent images, and we again use machine learning to identify the pathogen species for each milk curve, and then we map it back to the chip to count how many of each species. For example, the blue one with and without drug, the red one with and without drug. Then from the amount of DNA that you can quantify, you could say that bacteria A, because the amount are so different, that means the species are susceptible. While the red one, the amount are really close, then you could say that this is a resistant strain. This is an example of our nanoarray, and the fluorescent images after PCR. You will see the bright fluorescent spot that is the positive reactions after PCR, where the dark one are the negative wells, where it doesn't contain any DNA molecules at the beginning before the PCR. So this is the example. If we increase the temperature, if we have 2 different populations, you will see probably half of them already become dark, while the other one become dark later. meaning there are two different species in this sample. We have shown with the spike polymicrobial urine sample. We spike in two different bacterial species, and then we split them, incubate with drugs and without drugs, and perform machine learning, and then count the amount of bacteria. At the end, after comparing the amount of each milker, we could conclude that we detect there is a susceptible, Staph aureus, and resistant E. coli in the sample, the same as we expected. So the summary of this platform, meaning we propose a bacterial IDHT using microfluidic platform with this device. So now I am going to jump to another topic where we could use the same design of the device to evaluate human heterogeneity through DNA methylation from cell-free DNA. A quick recap of cell-free DNA is basically cell-free DNA, meaning it's a piece of DNA that are flowing in our body, in your bloodstream. And where does it come from? It's basically shed from tissue or dying cells, including tumor cells, which can be obtained very easy to just simply blood draw, right? And these pieces of DNA contain cancer-specific biomarkers such as mutation, methylation, and etc. What we are interested in is DNA methylation, which is one of the epigenetic silencing mechanisms. What it does is there is a methyl group that are attached to the cytosines on the CpG dinucleotides in promoter region. This can alter the expression of genes. So it has been found that aberrant DNA methylation patterns have been associated with majority of human cancer. Tumor suppressor genes are particularly important for maintaining cellular stability, and likewise their promoters are normally heavily protected against methylation. The presence of even a moderate methylation in certain regions of the promoters have been shown to affect genomic stability and potentially result in development of cancer phenotype. And the majority of tumors are heterogeneous in nature, which means they're often composed of different populations of cells. We're showing in different colors here. Different colors meaning it contains different molecular properties. These properties modulate their response to therapeutic intervention as well. Typical tissue biopsy only sample a small portion from the whole tumor, which fails to represent this heterogeneity. So we believe that investigating cell-free DNA allows for a better snapshot of cellular population from the entire body, including metastasis. It is promising, but there are some challenges. First is very low amount of cell-free DNA in the blood, and typically only 1 in 1,000 or even 1 in 10,000 that is a tumor DNA. Meaning in the 2 ml of blood, there are only a few copies of DNA, and really, really rare that will be a tumor DNA. So detect this copy is like we are trying to find needles in a haystack. And what we try to do is even try to differentiate the colors of the needles in the haystack. So that is mean the heterogeneity. The state-of-the-art techniques are methylation-specific primers or sequencing based like next generation sequencing. For the MSP, they are mainly used for detecting fully methylated. For example, we have 5 CpG sites. These MSP will detect only if these all 5 locations are methylated. But heterogeneity means some of them probably not methylated, so this MSP might not find those copies. Next generation sequencing, as all we know that it's quite expensive, especially if you want to do it very deep sequencing. So here we propose a method called dreaming or discrimination of rare acute alleles by NOS. It's capable of detecting the DNA methylation at single copy sensitivity and single CPG sites. It basically contains three parts: DNA extraction, conversion, digitization, and the analysis with HRM. The first part is about the conversion. Conversion means there's a bisulfite treatment with the process to change unmethylated CPG to, it will change cytosine to thymine, while the methylated cytosine will remain the same. This allows us to be able to analyze this using HRM, as the unmethylated will melt earlier than methylated copies. Here we show that we can detect different degree of methylation heterogeneity. The circle on the top right, the black circle meaning it's unmethylated, and the field circle means that it's methylated. So the blue one is all methylated copies that melt the highest in this plot. And we have the color-coded showing the level of methylation. We apply this method to detecting this example from non-small cell lung cancer stage 3 in P14 gene. We could detect the green and the orange one, which means it's the low or the middle range of methylation. And we apply this method to detect in other stages of cancer as well. And we show that we compare to the striped box, those are the conventional MSP methods. We can detect a lot more copies, especially the ones that are lightly methylated. So this method can also be performed on our microfluidic device. And this work has already, my lab mates already published this work about doing this methylation profiling using the microfluidic chip. So that's all of my talk, and I would like to acknowledge the lab, Dr. Jeff Wang from Johns Hopkins, and all the lab members that support this work, and also the funding agency, Royal Thai Government Scholarship, and NIH. Thank you for your attention.
Okay, thanks to the sharing from Dr. Athamanolap. So we are open the question for the audience.
Here, may I ask a question? Can you hear me?
Yes.
Okay. Very nice talk. So I was also working on some similar work in my lab. So I'm wondering, in your work, how long does it need to take for the AST to take effect? So overall, the turnaround time.
The overall, right. So thank you for the question. So the overall turnaround time, if we use a chip, it's probably about 4 hours. The incubation can be as short as half an hour. Mm-hmm. It also depends on the species as well, but like sample processing, preparing the reagents, the overall time is about 3 to 4 hours.
I see. And then the second question is for in a preparation, you need to treat your bacteria with antibiotics. So typically, how long should they be treated? And will it be different if you treat them with the different times? I'm not sure if you tried that in your experiment.
Right. Yes, that's a good question. So actually, it really depends on the species of the bacteria as well. We have tried like E. coli, we could see the very fast results. E. coli, Staph aureus, we could see the different amount even within half an hour or 1 hour. But for some species that are growing slower, then we probably need about like 4 hours.
Okay, but in general, if you have no idea about the bacteria in your sample, then how are you going to determine how long we have to treat the sample?
Right. Currently, we claim that we want to do 4 hours for the list of bacteria that we have already tested. we test a panel of the bacteria that we know that within the 4 hours we would see the difference in the result. But
Okay.
for new species, then we have to test to see how
see.
fast it's growing firstly.
I see. Thank you.
Okay, thank you for answering of the from Dr. Athamanolap. So currently, because time limit, I have to close this section, the webinar now. So before I close this webinar, I have a certificate to the Professor Wu. And then after we will send the same paper by the email. So please.
Okay, thank you.
Okay, Professor, Dr. Athamanolap, can you hear me?
Yes.
So we still have some question. Can you help me to answer this question?
Yes, I am about to type the answer now. Right. So the different result between two bacteria, basically different bacteria we will have to refer to its own reference. So we first need to identify what bacteria it is, and then the range, the difference of resistance will be different depending on the type of bacteria. So if you know it's E. coli, then you need to use the threshold of the E. coli to see if it's resistant or susceptible. For Staph aureus, you need to use another threshold, for example. So the result has to be used combined, like ID and AST.
Okay, thank you. This is Josh. So I enjoy your talk very much. So I also have a question regarding to your second part. So does your system can also detect, because like in the methylation, we know we have a monomethylation, bimethylation, and trimethylation. So and then for the heterogeneity of different cancer, they can have probably different methylation. So can you detect all these kind of mixtures?
So we have to pick the region of interest. We want to, that would be our dream, but then, right, we have to study first, like, which region that can be used for, like, lung cancer or different type of cancer will have different regions of methylation that we will need to look at.
Okay, thank you.
Okay. Okay, thanks for your participation. So see you next month.
Thank you.